Welcome to this NPTEL online certification course on biological process design for wastewater treatment. So, today we are going to learn regarding the bacterial growth process and its kinetics. So, in today's lecture we will start with the first studying the biological oxidation of carbonaceous matter and how it progresses. So, already we have studied in previous lectures little bit regarding the process of the formation of different biomasses, in particular the bacterial growth process. So, we will continue with the same. So, we can easily understand that during the aerobic reactions for the stabilization of organic matter in a closed system like in a bottle such as the BOD which was used in the BOD test, the sequence in which the mechanism of biological oxidation occurs follows two steps. First step or the initial stage includes the predominance of synthesis or anabolism reaction. So, in this reaction the organic matter which is present in the wastewater is used by the microorganisms for their metabolic activities of growth and energy conversion. Therefore, the prevailing activities related to synthesis, these are the first step towards the activity of the synthesis, and this phase results in oxygen consumption like the one reaction which is shown here, and then increase in the population of the microorganism which is shown here, and certainly during the reaction we have CO2 which is getting emitted and we obtain energy during the process. So, the oxidation takes place of the organic matter and we get energy out of this process as well as some amount of microorganisms get formed. In the subsequent stage, the second stage, we have endogenous respiration or catabolism which happens. So, the when organic matter originally present in the wastewater gets mostly removed, then there is a predominance of this second phase of oxidation. So, in this particular stage the microorganism population is at their maximum, and due to the low availability of substrate because all the organic matter has already degraded, the main food source becomes the cellular protoplasm itself. Therefore, in this phase there is a predominance of auto oxidation mechanism or endogenous respiration. So, this in during this process the microorganisms themselves actually they oxidize, and then we have CO2, ammonia and H2O which gets released along with the energy. In a system with mixed culture such as reactors used in the biological wastewater treatment, there are microorganisms with different growth and decay rates, and therefore, some microorganism species can be in one stage of synthesis, whereas the other may be in the second stage or endogenous respiration stage. So, this is possible that they may have different growth phases because we are using mixed culture. The representation which has been given here, it is only the average condition of heterotrophic microorganisms in the reactor. The total oxygen consumed in both phases like in the first phase anabolism and catabolism, it can be defined as the ultimate oxygen demand. So, we have studied the BODU which is the total oxygen consumed in the both phases. The addition of previous two equations leads to the simplified equation of this form. So, we can combine the previous equation 1 and 2 to write the simplified equation for the organic matter, and the value of theoretical oxygen demand, already we have studied regarding the theoretical oxygen demand. So, if we calculate the theoretical oxygen demand for this particular reaction and based upon the stoichiometry of this reaction, we will be finding that this will differ from the BODU value. So, why this is happening? And this is so because the living as well as the dead bacteria which actually serve as food for the higher organisms, during their transformation or in the new oxidation reaction that occur, a certain fraction of these organic matter they are always resistive to the biological attack. So, they do not oxidize and as such they remain in the water itself. So, this fraction actually which does not get oxidizes causes the deviation from the theoretical oxygen demand value and the BOD value. So, THOD value will be higher, but BOD value ultimately in the water will be lower, and that can be identified by the various tests etcetera, and then further calculation. So, this is there. Now, if we actually see that what is the progress of oxidation of carbonaceous matter, we can represent the curve using this type of figure, and here there are the growth is given with respect to three different things, but one is that how the cells or bacteria are increasing. So, we can see that they are increasing like this, and so here we have two sections. One is the synthesis section where the maxima has been reached, and after the maxima has been reached we have endogenous metabolism or endogenous respiration which happens. So, now the bacteria themselves are being utilized as a source of energy for the other bacteria which are there in the system. Now, substrate will certainly get substrate or the organic matter, it decreases with the increase in time and it is continuously decreasing. The oxygen consumption is continuously increasing and it will maximize to a certain value which is BODU. So, that we studied. So, this is the total oxidation of carbonaceous matter along with the time and the time progress of the oxidation of the carbonaceous matter. So, this can be represented by this figure. Now, going further, already since we have studied, we will be concentrating on the bacterial growth phase now first, and the synthesis, how we can represent the bacterial growth etcetera. So, that we will be studying in this part of today's lecture. The synthesis phase, we have two phases we have studied, the synthesis phase and the endogenous respiration phase. The heterotrophic microorganisms, they use organic matter as a form of condensed energy for their various metabolic processes including growth and reproduction. So, with the use of oxygen and or any other electron acceptor, these organisms they oxidize the organic matter with the production of more cellular matter. So, that means we have growth and reproduction of the microorganism and energy release. So, this is the synthesis phase we have already studied. During the endogenous respiration phase, if the substrate is available in the medium which will become less because we have already peaked and the synthesis phase is virtually over. So, under those conditions the substrate which is available in the medium that becomes scarce. So, under that condition the organic matter is already removed and the microorganism need to find organic matter from other sources. So, the main substrate which is available for the cell to use under this condition is their own cellular protoplasm. So, they start eating themselves. So, in this stage the balance is negative because reduction of cellular matter is happening or the bacterial concentration in the medium is decreasing. So, this is called the endogenous respiration stage. So, this is there. Overall the bacterial growth curve which was earlier given also, it can be represented like this. So, we have the synthesis phase up to which the maxima is occurring and then we have endogenous metabolism which is occurring. Now, we can divide the bacterial growth curve into different phases and these phases are depicted here, are listed here. So, these are the first phase is called lag or adaptation phase. Then we have logarithmic phase where the exponential growth happens. Then we have stationary phase or the in a way growth and decline phase both are equal. So, we have virtually the stationary phase and then finally the endogenous respiration stage where the bacteria starts decreasing continuously with time. So, this is the bacterial growth curve which is we can see and observe if we do some wastewater treatment or growth of bacteria inside a batch reactor. So, this will be the curve that will be obtaining. Now, different phases are defined here. So, the lag phase actually it is the period during which the enzymatic adaptation of the bacteria to the new substrate happens. So, this phase can be reduced in the case of typical domestic sewage in which the bacteria has already acquired the necessary enzymatic equipment. So, this lag phase is with respect to acclimatization of the bacteria in the new conditions with respect to substrate, with respect to temperature, etc. After initial lag phase, we have experimental growth phase. So, here the cells divide themselves at a constant rate, and we have on, if we plot this on the logarithmic scale, the number of cells grow linearly. So, thus this is exponential growth phase, and as there is an excess of substrate, that means the organic matter which is present in the medium is very high. So, thus the growth rate reaches its maximum, and with the only limitation by the microorganisms capacity to process the substrate. So, depending upon that how quickly microorganism process the substrate, it will be there. So, we have abundance of organic matter or substrate present in the system during this phase. Along with the maximum growth phase, there is also the maximum substrate removal rate also, because the microorganisms are growing as well as the concentration of the organic matter is decreasing the fastest possible. Now, after this exponential growth phase, we have stationary phase. So, in this phase the food in the medium starts to decrease and starts to become scarce, and the bacterial growth rate becomes equal to the death rate, and so the number of cells in the this phase virtually remain constant, and after the stationary phase we have decline or decay phase. So, in this phase the substrate availability in the medium is reduced and the endogenous respiration prevails, and the bacteria are forced to use their own cellular protoplasm as a substrate source. Now, in this condition the bacteria start using their own cellular protoplasm as organic matter to get the energy which is required for their sustenance. So, nutrients of the dying cells diffuse into the medium and serve as a food to the other cells, and the death rate is exponential and is constant and leading to a straight line on the logarithmic scale. The importance of this whole phases is that it represents the growth regarding a single population of microorganism growing at the expense of single type of substrate. However, in reality the biological reactor of a sewage treatment or wastewater treatment, there are variety of microorganisms which are metabolizing a number of compounds. So, different types of organic matters are present, different types of microorganisms are present, hence there will be a overlapping of various curves of different forms and types, and thus they will be developing at a different time. So, there the growth, the overall curve may be different as compared to the one, and will be having a more stable the stationary phase will be more there in the actual condition. Now, what are the observed tendencies to use these concepts in the sewage treatment plant? So, how to, what are the different how to use this curve in the sewage treatment plant, this is little bit discussed here. So, if very high loading systems are there, that means the substrate is very high. So, in the exponential growth phase the substrate availability is high. So, this indicates that the concentration of substrate, example like BOD in the effluent will also be somewhat high, and thus the majority of sewage systems do not operate in this phase. So, very high loading systems are not very common because BOD is generally average. Now, in the second case we have high loading system in place of very high, now we have high loading system. Example, the conventional activated sludge process, high rate tickling filter that we are going to study later on in detail. The concentration of substrate in the effluent is lower, but the cellular mass has a high organic fraction requiring the separate stabilization of the excess sludge. So, we have to condition the excess sludge, we have to separate out the excess sludge, so that we can maintain a particular cellular mass in the activated sludge process or high rate tickling filter. Due to the high load, the volume required for the reactor is smaller than the low loading system that are possible. Now, there could be low loading systems where BOD is less. So, for this the activated sludge system of the extended aeration variant is used or low rate tickling filters are used. The reasoning for this system is to supply a minimum quantity of substrate to the organism in order to stimulate the endogenous respiration. This leads to self metabolism of the microorganism, that is, they undergo a digestion of the cellular mass in the reactor itself, and besides the partial stabilization of the cellular mass, the concentration of the substrate in the effluent is also low. So, under this low loading system, the system is operated under a condition where some amount of endogenous respiration is happening. So, we do not have to that much care of the excess sludge, otherwise in the high loading system we have to take care of the excess sludge, because the system is still operational under the exponential growth phase and we have to separate out the excess sludge continuously. Thus, in the low loading system, the volume required for the reactor is larger than for the high loading system, but we do not have to take care that much of the excess sludge. So, this is the good thing. Now, we are going further and we will try to understand the kinetics of the bacterial growth. Now, we have studied the phases, now we will try to understand the kinetics of the bacterial growth. Now, one term is called specific gross bacterial growth. So, the bacterial growth can be expressed as a function of bacteria concentration at a given time in the reactor. The net growth rate is equal to the gross growth rate minus the bacterial decay that is possible. So, the growth rate of a bacterial population is a function of its number, mass or concentration at a given time. So, thus we can write the if we are assuming that x is the concentration of microorganism in the reactor and which is like suspended solid or the volatile suspended solid. So, they actually this SS or VSS they represent the concentration of the microorganism in the reactor. So, already we have studied regarding SS VSS earlier during the wastewater characterization. So, this if this is the x, so the growth rate of x, that means the dx by dt as per this will be proportional to the concentration itself, and the proportionality constant is called specific growth rate. So, this is there the time where t is the time in days or any other appropriate unit which is which can be used. Going further, in this previous equation, when if we integrate this equation and it will assume a exponential form, that will be having a equation which is like x is equal to, if we assume something x e x to minus k t. So, this will be something of this form nu t. So, this will be this form, and if we write the log form of this, so this will become a straight line. So, that is like if log x is equal to log x 0 minus nu 2. So, this is the typical bacterial growth curve it will be. The growth rate is for without limitation of the substrate. However, the bacterial growth as a function is a function of availability of substrate in the medium. So, it is not, we should not use this particular equation, because the x, the growth rate of x is actually a function of substrate also. So, when the substrate is present at a low concentration, the growth rate is proportional low, and in sewage treatment the carbonaceous matter is usually the limiting growth factor. So, how much amount of organic matter is present or not present, depending upon that the growth happens. So, under this condition the specific growth rate nu must be therefore expressed as a function of the substrate concentration. So, this was the idea that nu should be function of substrate concentration. So, Monode presented this relationship according to the following empirical equation, that nu is equal to nu max S upon Ks plus S. So, here nu max is the maximum specific growth rate, this is, and its unit is per unit time, here like per unit day. Then S is the concentration of limiting substrate or nutrient gram per meter cube. Ks is the half saturation coefficient which is defined as the substrate concentration for which nu is equal to nu max, and this equation can be plotted like this, the one which is shown here. So, later on, so this if we plot nu, this can be represented by this equation where the nu is continuously increasing and it goes to nu max. So, this is there. Now, we will try to understand that how this growth happens. So, the kinetics represented by Monode equation. So, we will be trying to discuss this Monode equation now. In the case of highs, in the case where the substrate removal reaction is happening and when the concentration is still high and it is not limiting, that means S value is very high and it is not limiting. Under that condition, the global removal rate approaches the 0 order kinetics. So, in this case it is not dependent upon the substrate. So, we have this is the condition where high concentration of limiting substrate is there. The reaction this growth will always be at the nu max. So, this is the condition when the consumption of the with the consumption of the substrate the reaction starts to decrease and the characterizing a transition or mixed order regime that is represented here in this condition in between condition. So, this is like mixed order regime. After that when the substrate concentration becomes low, the reaction order starts to be limited by the low availability of the medium. So, in this condition the reaction kinetics approaches the first order kinetics, which is shown here in this figure. So, we can see the reaction order becomes less equal to 1 and low, and this is true for low concentration of limiting substrate. And these any of these condition may happen depending upon the relative values of S and Ks, which was defined earlier. Now, what is the relative? There are different conditions already we see. So, when the relative substrate concentration is high, so S is much greater than Ks. So, under that condition nu becomes equal to nu max, and this reaction follows 0 order kinetics in which the reaction rate is independent upon of the substrate concentration. So, this will happen more in the domestic sewage treatment, and this situation tends to occur at the head of a plug flow reactor where the substrate concentration is still high. So, we will in the next few lectures we will be studying regarding plug flow reactor, CSTR, etc. Now, when the relative substrate concentration is low, that means S is much less than Ks. So, under that condition the equation becomes nu is equal to nu max S upon Kx, and now we can write nu max upon Ks as another constant K, and thus the equation actually changes to nu is equal to Ks. So, that means now the growth is function of substrate. So, the growth rate is proportional to substrate concentration and the reaction follows first order kinetics, and this will happen in the treatment of domestic sewage in a complete mixed reactor, and generally the activated sludge process etc. are like a complete mixed reactor. So, the growth rate in the complete mixed reactor follows first order kinetics, and it will be the true for the activated sludge process etc. So, this is the overall curve is the one we have nu is equal to mu x, reaction order is 0, and when the low concentration of limiting substrate is there, the reaction order is 1, and this is the condition. So, this is the growth. Now, in the laboratory the curve of specific growth rate as a function of concentration of limiting substrate can be constructed, and the value of Ks and nu max can be extracted. So, in domestic sewage treatment values of Ks and nu max in the following ranges have been reported in the literature, like in Metcalf AD for aerobic treatment the nu max value has been reported in the range of 1.2 to 6 per unit day. Then similarly, the Ks value is from 25 to 100 milligram BOD 5 per liter, or Ks in terms of COD is 15 to 70 milligram COD per liter. For anaerobic treatment, the nu max value has been reported in the range of 0.4 to 2 depending upon whether acidogenic organisms are present or methanogenic organisms are present. Similarly, the Ks values are in the range of 50 milligram COD per liter to 200 milligram COD per liter depending again on the acidogenic or methanogenic organisms are present or not. So, there are different types of, there are certain limitations of Monode equation as well as the advantages are also there. So, Monode equation, the limitation is that the Monode equation is not based upon the theoretical principles. It is essentially empirical in nature, and this was derived for a single organism metabolizing on a single substrate. However, in wastewater treatment there are a number of multiple population assimilating in a multiple substrate. So, thus we have, this is the major deterrent in the use of Monode equation for the wastewater treatment. Now, advantage is that the Monode equation structure permits the representation of continuous form of the range of variation between extremes of lack and abundance of nutrients in the medium. So, we can easily establish that, and therefore, depending upon the value of S in terms of BOD or COD, the Monode equation can represent approximately the kinetics of 0 and first order as well as the transition between them. So, it is very easy to represent the kinetics and the transition in between them between 0 and first order using the Monode equation. I will take one example before closing this section. So, here it is being asked to express the growth rate as a function of nu max for the following condition. When the domestic sewage, the substrate concentration is S is 300 milligram per liter, and in another condition the for the substrate is 10 milligram per liter, and it is being suggested that to use the Ks value of 40 milligram per liter for both the cases, and in the third case the S value is assumed to be 10 milligram per liter and the Ks value is suggested to be 0.2 milligram per liter. So, for first case if we write, the nu value will be 0.88 nu max. So, we are just substituting the value. So, in this case we are finding the value to be 0.88 of the nu max. So, that means the growth rate is around the maximum value. In this condition in which S is large in comparison with Ks, we can see the S is much larger than the Ks, the growth rate is close to nu max. This is, that is, means that there is a greater availability of the nutrient and the population present is in a high growth rate phase. The reaction is appropriately of 0 order. Now, this condition will not be very frequent in the treatment of domestic sewage and occurs only in some cases. Now, for the second case, when the S is 10 milligram per liter, it is 0.2, the nu is found to be 0.2 nu max. That means, as the S is small in comparison with Ks, the growth rate is much lower than nu max, and this indicates that there is not much availability of the limiting nutrient in the medium. So, this is there, and this will be possible in the complete mixed reactor like activated sludge process, etc. For the C case, when the S is 10 milligram per liter and Ks is only 0.2. So, again we are finding nu is equal to 0.98 nu max. So, again though the concentration of S is the same as in the item B, but since in this case glucose for the glucose Ks is very low and it has high affinity, the bacteria have very high affinity, the denominator of the expression is practically equal to S itself, and in this case the nu is almost equal to nu max. So, consequently there is a high availability of substrate and the growth rate is very close to the maximum. So, this is the, so today we have learned regarding little bit of the growth kinetics and the Monode equations and how to use the Monode equation under different conditions of substrate concentrations, and whether, and we also learned that what is the impact of substrate concentration on the growth curve as well as the value of Ks. So, with this we will end today's lecture. Thank you very much.